What we need to look at now is the way that the smallest blood vessels of all work, that is the capillaries, the microscopic circulation, the circulation on a very small scale. So let's start off with a diagram of a capillary. Capillary, remember, is made up of squamous epithelial cells, very thin cells. So capillary made up of very thin, flat cells. These are the individual cells comprising the wall of the capillary. This is a capillary wall I'm drawing here. And a useful way to think about this is that there's very small gaps between some of the cells that make up the capillary. And we're talking about a capillary in the systemic circulation. What we've got, of course, is a continuous tube. Obviously, this will be in 3 dimensions. It's a tube. I'm only drawing it in 2 dimensions. So here we have the cells that make up a capillary. Now this will be the arterial end of the capillary here, with oxygenated blood entering. And actually capillaries are so small that red blood cells very often only go through the capillaries one at a time. So very small structures we're considering here. There's a red cell, darker on the outside, lighter on the inside because it's a biconcave disc. So red cells passing through this capillary just one at a time. So oxygenated blood entering the arterial end of the capillary. Of course it would be longer than I've drawn it, but it's still true to say that deoxygenated blood would leave via the venous end of a capillary. The blood enters the capillary via an arteriole, and the blood leaves the capillary via a venule. And there's millions of these capillaries in the tissues. So I don't know if we can actually focus in on my fingernails there, but yours will be exactly the same if you look at your fingernails. You can see that fingernails are pink. But if we actually press on the fingernail like that, you can see it goes white. Now the reason it goes white when you press it is because the pressure of the nail on the underlying capillaries is actually emptying the capillaries, and you're seeing the white coloured tissue. Then when we let go, the blood supply fills the capillaries up again. It's called capillary refill, and you can see it goes pink again. So the pink colour is caused by the blood perfusing through millions of capillaries. Now round about the capillary, of course, we have tissue cells. These are the cells which comprise the tissue which is being perfused. Tissue, you remember, is a group of similar cells together with its connective tissues. So here we have the cells which are actually being supplied by this capillary. But notice the blood is not actually in contact with the tissue cells. But round about the capillary, the tissue cells are bathed in tissue fluid. So the cells of the tissue bathed in tissue fluid. Now these cells are actively metabolizing. That means they're using up oxygen and producing carbon dioxide. So that means when the blood arrives from the arterial end of the capillary, and remember when the blood arrives in the arterial end of the capillary, it is going to be high in oxygen and low in carbon dioxide. So you have a situation where the blood here is high in oxygen, whereas the tissues, because they've used oxygen up, are relatively low in oxygen. That means there's more oxygen in the blood in the capillary than there is oxygen in the tissues. What that means is that we have a diffusion gradient between the blood and the tissues. Now, diffusion is the tendency for substances to intermingle until their concentrations become equal throughout. So because there's more oxygen in the capillary than there is in the tissues, there'll be a movement of oxygen from the blood, from the capillary blood, into the tissues. So there will be diffusion of oxygen from the blood into the tissues. That's good, of course, because it means the tissues are oxygenated. Initially, the oxygen will just diffuse into the tissue fluid, and from the tissue fluid it will diffuse into the cells themselves. So there's a diffusion of oxygen down its diffusion gradient from the blood in the capillaries into the tissues. And of course, it's only in the capillaries that the vessel walls are thin enough to allow diffusion to take place. Diffusion does not take place across the walls of the arteries and the veins. The walls are simply too thick. Now, we've said that the blood arriving in the arterial end of the capillary is going to be low in carbon dioxide. However, the tissue cells, because they're actively metabolizing, means that they're going to produce carbon dioxide as a waste product of tissue respiration. You might remember the equation that goes on in the mitochondria. Glucose plus oxygen gives carbon dioxide plus water plus energy. So they're using up oxygen, generating carbon dioxide. Because the cells are generating carbon dioxide, that means that the amount of carbon dioxide dissolved in the tissue fluids is going to increase. The arterial blood arriving in the arterial end of the capillary is low in carbon dioxide. So we have a situation where there's high levels of carbon dioxide in the tissues and lower levels of carbon dioxide in the blood. That means there is a diffusion gradient between the tissues and the blood. So there's more carbon dioxide in the tissues than there is in the blood. So carbon dioxide will diffuse down its diffusion gradient from the tissues into the blood. In other words, in simple terms, we get net movement of carbon dioxide from the tissues into the blood. So carbon dioxide moves from the tissues into the blood. Oxygen moves from the blood into the tissues. So as the blood goes through the capillary, it gives up its oxygen and it accumulates carbon dioxide. So the blood arriving at the arterial end of the capillary is high in oxygen and relatively low in carbon dioxide. The blood leaving at the venous end of the capillary is going to be low in oxygen because it's given it up to the tissues, and high in carbon dioxide because it's collected blood from the tissues. So there's this exchange of gases across the wall of the capillary to the tissue cells. Now, as well as an exchange of gases, there's exchange of other materials. For example, as we've said, the cells are using up glucose. So when the blood arrives, the blood is going to contain glucose, and the glucose will diffuse from the blood into the cells that have used the glucose up, providing there is a diffusion gradient for that to occur. So if the cells use up the glucose, more glucose will diffuse from the blood into the cells. And it's the same for other nutrients that the cells require. So there'll be transfer of amino acids from the blood to the tissues, to the cells of the tissues. There'll be transfer of vitamins from the blood to the tissues, to the cells in the tissues. So the cells are also supplied with nutrients from the capillary blood by diffusion, mostly by diffusion of these products across the capillary wall. Now, as well as that, these cells are also producing waste products of metabolism, often containing nitrogen. So there'll be diffusion of these waste products, again from the cells into the tissue fluid and back into the blood. So net diffusion of waste products of metabolism from the tissues back into the blood, where that can be taken away and the waste products can be processed by the kidney or the liver or whatever is processing that particular waste product. So all the tissues of the body are oxygenated and nourished via the blood that perfuses those tissues, which is travelling through the capillaries in those tissues. All the waste products are taken away from the tissues by the circulation in the capillary blood through that tissue. What we're going to do now is carry on and look at another aspect of capillary physiology, that is the formation and reabsorption of tissue fluids. And again, we'll use a diagram to help illustrate this. Now we know that a group of capillaries is supplied with blood from a microscopic blood vessel called an almost microscopic blood vessel called an arteriole. So the arteriole takes blood into a group of capillaries. I'll only draw 3 to keep the diagram fairly straightforward. But I think you can see what's happening here. We've got blood going into the arteriole and into this network of capillaries. And the capillaries are drained by a very small vein called a venule. And in the middle we have the capillaries. So an arteriole taking blood in, the blood circulating through the capillary bed, and the blood being drained via the venule. And of course, as we know, this capillary is going through a tissue, and the tissue contains tissue cells, the cells which comprise the tissue. Now, we know that the pressure of the blood in the arteries is higher than the pressure of the blood in the veins. And it's the same in arterioles and venules. So that means this pressure drop between the arteries and the veins partly occurs as the blood goes through the capillaries. So the blood at the arterial end of the capillary is at greater pressure than the blood at the venous end of the capillary, and this is very significant. So the blood at the arterial end of the capillary has a pressure of approximately 32 millimeters of mercury. So here at the arterial end of the capillary, approximately 32 millimeters of mercury pressure. About 32 millimeters of mercury pressure. Whereas at the venous end of the capillary, the pressure's lower, the pressure's dropped, and the pressure here at the venous end of the capillary is probably something like 12 millimeters of mercury, quite a bit lower. So, arterial end of the capillary, blood at fairly high pressure, 32 millimeters of mercury. Venous end of the capillary, blood at fairly low pressure, 12 millimeters of mercury. Pressure drop as the blood goes along through the capillaries. Now, the blood contains plasma, and the plasma contains plasma proteins. Proteins are big molecules. Big molecules exert an osmotic suction. So the plasma has an osmotic pressure. And the osmotic pressure of the plasma is about 25 millimeters of mercury. So let's just put that figure down as well. Osmotic pressure of the blood, of the plasma, about 25 millimeters of mercury. Now, can you see now that the blood pressure at the arterial end of the capillary is greater than the osmotic pressure at the arterial end of the capillary? Now, the blood pressure is going to tend to force fluid out from the capillaries into the tissue spaces through those small gaps in the capillary wall that we drew before. So at the arterial end of the capillary, because blood pressure or hydrostatic pressure is greater than osmotic pressure, remember the osmotic pressure is trying to suck the water in, because the blood pressure is greater than the osmotic pressure, there's a net pressure of 7 millimeters of mercury tending to exude the water. So tissue fluid is formed at the arterial end of the capillary because hydrostatic pressure pushing the fluid out is greater than the osmotic pressure sucking the fluid in. So we get formation of tissue fluid. It's very fortunate that this does happen because the tissue fluid is essential to bathe all the cells of the body, otherwise they would dehydrate. The tissue fluid is also essential to facilitate the diffusion of oxygen from the blood through the tissue fluid to the cells. Tissue fluid is also essential to facilitate the diffusion of carbon dioxide from the tissues through the tissue fluid back to the bloodstream. Tissue fluid is also essential to facilitate the diffusion of glucose from the blood through to the cells of the body. And of course the removal of waste products from the cells, from the tissue fluid, back into the bloodstream. So tissue fluid, sometimes referred to as interstitial fluid, is absolutely essential. And it's formed because at the arterial end of the capillary, hydrostatic pressure, or blood pressure, exceeds osmotic pressure. But as we've said, there's a pressure drop as we go along the capillary. So at the venous end of the capillary, before the blood drains into the venule, the pressure of the blood here is only 12 millimeters of mercury. But what is the osmotic pressure of the blood inside the capillary? Well, that remains at 25 millimeters of mercury. So now the pressure drawing water in is 25 millimeters of mercury, and the pressure exuding water out is only 12 millimeters of mercury. So this means that the osmotic effect sucking water back into the capillary is greater than the hydrostatic effect pushing the water out of the capillary. That means there's a net movement of tissue fluid from the tissue spaces back into the blood, being sucked in by the osmotic pressure of the plasma. So at the venous end of the capillary, because osmotic pressure exceeds blood pressure or hydrostatic pressure, there is reabsorption of tissue fluid. This means tissue fluid is formed at the arterial end of the capillary, circulates through the tissues, and is reabsorbed back into the venous end of the capillary. So there's movement of tissue fluid over the cells that comprise the tissue with fresh tissue fluid. So fresh tissue fluid forming at the arterial end, going over the cells, being reabsorbed at the venous end. So as well as being circulation of blood through the capillaries, there's also circulation of tissue fluid derived from the plasma, returning to the plasma, past the tissue cells. What we're going to look at now is the lymphatic system, and particularly the lymphatic drainage of the tissues, lymphatic drainage. Now, we've already discovered that there's an arteriole group of capillaries. Again, we'll use the same sort of diagram as before. And a venule. And we've said there's formation of tissue fluid at the arterial end of the capillary and reabsorption of tissue fluid at the venous end of the capillary. So we've got tissue fluid accumulating here in the tissues. We've got the cells of the tissues. And tissue fluid accumulates. Now, not all of the tissue fluid drains or is absorbed back into the venous end of the capillary. There's an additional system involved, and this is the start of the lymphatic system. So in the tissue spaces all over the body, we have blind-ended lymphatic capillaries. They tend to be a bit thicker than the blood capillaries. So blind-ended lymphatic vessels, large networks of them. And again, a bit like veins really, forming larger vessels. So infiltrating all of the tissue spaces, we have a separate drainage system. These are called lymphatic capillaries. And excess tissue fluid drains into the lymphatic capillaries. Once in the lymphatic capillaries, this fluid moves down into the larger lymphatic vessels. And these larger lymphatic vessels again join larger vessels until you get larger still lymphatic vessels draining the areas of the body. Now, it's true that these lymphatic capillaries do drain to some tissue fluid. But as well as that, there's some loss of proteins from the capillaries, larger molecules. And the lymphatics are vital to drain those larger molecules. Because if large protein molecules are left in the interstitial fluid, that would increase the osmotic pressure of the interstitial fluid, of the tissue fluid. So to prevent that occurring, any proteins which are lost from the bloodstream are also drained via these lymphatic vessels. So fluid and waste proteins, unwanted proteins, go into the lymphatic ducts, and these drain into gradually larger and larger lymphatic vessels. As well as that, these can remove any infection from the tissues. So for example, if you've got an infected finger, then sometimes you can see red streaks going up the arm, which are infected lymphatic vessels. And these lymphatic vessels carry the blood, in the case of the arm, up the arm, and eventually drain into the lymph nodes, which are under the arm, the axillary lymph nodes. Now, different parts of the body are drained by different lymph nodes. So the legs, for example, are drained by lymph nodes in the groin. The arms drained by lymph nodes in the axilla. The back of the mouth and the tonsils are drained by lymph nodes underneath the mandible, under the submandibular lymph nodes. Because you can feel these swollen up when you've got a sore throat, because they're filtering bacteria from the tissue fluids, from the tissue spaces. Now, the tissue fluid, here we've called tissue fluid or interstitial fluid. When it enters the lymphatic vessels, it's fair to call the fluid lymph, when it's actually in the lymphatic vessels. So lymph fluid is drained in these lymphatic vessels. And these lymphatic vessels carry the blood towards the lymph nodes. And they contain valves to prevent backflow of lymphatic fluid. So the valves are pointing this way around. So fluid can travel from there to there, but the valve will not let the fluid back again. So one-way flow of lymph is ensured by the valvular system in these larger lymphatic vessels. Now eventually several of these lymphatic vessels will drain into a larger structure called a lymph node. So we have several lymphatic vessels draining into a lymph node. These are still fairly small structures actually. There's located under your arm in the places we've discussed. Now, basically, a lymph node is filled with a connective tissue, sometimes called a reticulum or a net-like tissue. So it's got connective tissue in there, which is a reticular network. Reticular tissue in the lymph node. And the key thing about this reticular tissue is it acts as a place that houses the body's defence cells that defend us against infection. This is these are actually filters, really. They're filtering the tissue fluid and any bacteria that get through into the lymphatic fluid. And as well as filtering infection, lymph nodes can also filter malignant cells. So if there's a malignancy in part of the body and there's spread to the lymph nodes, then that is another stage in the malignant process. But the lymph nodes can actually hold down or slow down the spread of malignant disease. So we've got these lymphatic capillaries draining fluid into large lymphatic vessels, travelling all the way up the arm, eventually draining into lymph nodes, several lymph vessels draining into lymph nodes. Now, the vessels which carry lymph into a lymph node are referred to as the afferent lymphatic vessels. So afferent lymphatic vessels carrying lymphatic fluid into the lymph node, which has this reticular tissue inside it, this network tissue. But the lymph node is actually full of the protective cells. Basically, two types of protective cells. The first sort are the white cells that are particularly associated with the lymphatic system, the lymphocytes. So the lymph nodes contain large numbers of lymphocytes. The white blood cells associated with the lymph nodes, and there'll be B and T lymphocytes in the lymph node. As well as that, the lymph node houses phagocytes, cells which can eat bacteria. So the lymph nodes, large numbers of lymphocytes and phagocytes, both involved in protecting the body against infection. So we see that lymph nodes are made up of reticular tissue, lymphocytes and phagocytes. Collectively, we refer to this as lymphoid tissue. So the lymph nodes contain lymphoid tissue to filter the tissue fluids, the lymphatic fluid draining from the tissues, and to deal with any infection which is in the tissue fluids. Leaving the lymph node, there's another vessel. It's called the efferent lymphatic vessel, so the lymph is leaving via this vessel. And that goes on eventually back into the venous circulation. So let's just review this. Tissue fluid is produced at the arterial end of the capillary, reabsorbed at the venous end of the capillary. However, some of the tissue fluid, plus any rogue proteins which have escaped from the circulation, are drained from the tissue fluid into the lymphatic fluid, drained by the lymphatic capillaries, and that passes from the lymphatic capillaries into the larger lymph vessels. One-way flow of lymph is ensured by a system of valves inside these lymphatic vessels. Several lymphatic vessels will drain into a lymph node. The vessels which drain into a lymph node we refer to as efferent lymphatic vessels. In the lymph node there is a reticular framework, net-like tissue, which houses lymphocytes, B and T lymphocytes, and phagocytes. This tissue collectively is referred to as lymphoid tissue. Once the lymphatic fluid has been filtered through the lymph node, it drains out via the efferent vessel from the lymph node. Now, when you have an infection, lymph nodes can become swollen and painful because bacteria can collect in them. Then there's a battle between the lymphocytes, the phagocytes and the bacteria. The body's immune system will start to fight the bacteria in the lymphoid tissue of the lymph nodes, and that can make them become enlarged and swollen. That's referred to as a lymphadenopathy. But under normal circumstances, once the lymphatic fluid is drained from the lymph nodes via the efferent lymphatic vessels, it collects into still larger lymphatic vessels and ultimately is drained back into the venous circulation of the blood. And it drains back in via two places. It drains back in via the left lymphatic duct into the subclavian vein and via the right lymphatic duct into the right subclavian vein. So ultimately that completes the circulation and the lymphatic fluid drains back into the venous circulation. And there's about 2 litres of lymphatic fluid drained by the lymphatic system, and about 2 litres the same every day re-entering the venous circulation. So this is another example of circulation throughout the body.